So let's split that into its front half. Now we're down to a quarter of the original song. And the back half, the other quarter of the original song. And let's do that again. Let's use this and now quadruple it to make a song of the same duration as the original. And take this and quadruple it to make a song of the same duration as the original. Perform the preference testing again. If this hypothesis is true, she should like this one. Then finally we're down to where we can't split it anymore. We have this that we used to make one stimulus and this that we used to make another, and she reports to us her preference again. Now we can look at the density. If we're going to hypothesize that this is true, then we would expect that the density of the attractive element should be related to the degree of her response, indicating her perceived attractiveness. So here, if we look at the density of the attractive feature, in the original song it had a density of 1, then we split it in half and reassembled it. It took on a density of 2. We split it again, it took on a density of 4. We split it again, it took on a density of 8. With each division, which we can talk about the division from level 1 to level 2, or from level 2 to level 3. We can just represent this as level n to level n plus 1. At each division, we are doubling the density of this attractive element. And so if we were to plot the response to the preferred stimulus at song division n plus 1 versus the response to the preferred stimulus at song division n, then we would expect a doubling for each division and a slope of 2. So in this model, where there is a single sexy element, we would expect that if we were to plot our data in this way, we would get something with a slope of 2. For hypothesis 2, we can go a little bit more quickly, but we'll just talk about the original song, which contains 8 different elements. The complexity of song content is quite high. We have 8 different elements in this song. So we split it and use it to create new stimuli, 1, 2, 3, 4, 1, 2, 3, 4, versus 5, 6, 7, 8, 5, 6, 7, 8, etc. Split it, retest, split it, retest. We would expect this to be relatively close as far as her reported preference. Maybe this is 55%, this is 45%, or something like that. But it doesn't actually matter which of these she chooses to let us interpret our results. Because whichever of these two that she chooses, the density of the attractive feature, that is number of different things, has gone from 8 to 4. It's 4 here, and it's 4 here. When we split it again, it goes from 4 to 2. When we split it again, it goes from 2 to 1. So if we were to hypothesize that this is the explanation for female behavior, or female preference, then a plot of n plus the response to n plus 1 versus the response to n should yield a slope of 1/2 if the complexity of song content hypothesis is correct. And then last, we have the neuromuscular challenge, which would be like me clapping, and I would just be clapping, and each clap would be equally impressive. So each of these contains a blue dot, or a blueness, indicating that each of them is equally attractive. We can split it and perform all these tests, and the density of attractive features here is 8, and 8, and 8, and 8. So this thing persists throughout the duration of song if this hypothesis is true. So in model 3, we get a slope of 1. So these are our predictions. That we have the singular sexy element hypothesis, we have the complexity of song content hypothesis, and we have the neuromuscular challenge hypothesis. And our data lie here. That we get the best fit linear regression as this dotted line here, and it is different, statistically different than a slope of 2. It is statistically different than a slope of 1/2, and it is statistically indistinguishable from the slope of 1. Yes?
What if it was an equal mix of model 1 and model 2? They were both like sexy songs and complexity were jointly determining.
Let me make sure I understand your question. What if there are multiple aspects at play determining this? Yes, that's true. A blended feature would be difficult to detect here. But if it were a single element, well, clearly you appreciate the hypothesis. All right, so what we're going to do is we're going to look at this model 3 more closely, because it appears that there's something that persists throughout this song. I'll answer your question in the end by these additional tests. But we're going to suspect that there's something that's present throughout the duration of this song that makes it attractive. So what we're going to do now is we're going to take the female residing in our testing chamber, and we're going to play to her experimentally altered versions of her most preferred song type. And here is the original song, and we're going to change 2 things. We're going to change either the frequency or the pitch, and we're going to change the tempo. Because it could be that she just likes this male's tone of voice, so to speak. There's something spectral about his performance that she really appreciates. If we were to change the frequency by going down 200 cents, or do, re, mi, 2 notes, if you know da, na, na, like that's 200 cents there. You take each note interval and divide it into 100 pieces, these are cents. We've gone 200 cents. Now here we're going to pitch the frequency up, and so da, da, da. And here we're going to accelerate the song in tempo, and here we're going to decelerate it. We can see that most easily here. If we look at this absolute peak of this note right here, and then we come across, then we see that for the, excuse me, I'm talking about this note, I pointed to the wrong one. Then for the control condition, it was here, and if we go across to the pitch increased condition, it's here. And you can see here that the notes are of approximately, they are in fact the same duration, but they are of different frequency. And then for tempo, if we look at this last note especially, then if we take this and just move it up here to look at it, these 2 notes are of the same frequency, but they are of different duration. And our ears are better than our eyes, so I will hopefully play these songs for you. We'll start with the original song. Could you guys hear that? All right. So here's the original, then I'll go low, high. Can you hear that? Yeah, there's a pitch difference. Now for tempo, we'll have the original song, and then a slowed down version, and the accelerated. This is effectively avian auto-tune, give or take. So when we ran these behaviors, we said, okay, we're going to compare the natural song versus a changed song. And so the preference for an altered song would be up on this axis, and a preference for the natural song, or a dislike of the altered song, would be down on this axis. And when we looked at changes in note frequency, individual birds are plotted as the gray connected dots. And the mean at each point is shown by this circle, and the fact that it's open means that it is not significantly different from a model of 0. That is to say, there's no significant change in preference as a function of changing frequency. However, when we change the tempo, then we got these data. And again, individual birds are gray. The means are shown in blue. Filled symbols indicate significant differences. So the linear regression tells us there's some systematic change with respect to the change in tempo, but not all birds agree. Some birds think it's more attractive and other birds think it's less attractive. And when we broke that apart as one group of birds that think that accelerated song is more attractive and others that think that accelerated song is less attractive, then we got one set of birds who report to us that an accelerated tempo makes a song generally more attractive, a decelerated tempo makes it less attractive. And we get another set of birds that are telling us that intermediate changes in tempo have little or no effect on their preference, but an extremely accelerated condition, in fact, makes this less attractive. So here what I've plotted is relative tempo. I've changed it by 10% or 25% or 50%. And that could be a change from 4 notes per second. A change of 50% could be a change from 4 notes per second to 6, or it could be from 12 to 18. That's a relative change in tempo, and the 12 to 18 would be a change in absolute tempo. So is it simply that the fastest thing this female has ever heard is the most attractive thing she's ever heard? Might absolute tempo be the answer? And to address that, we took our original data that I just showed you, with its regression here, and we then looked at absolute tempo. So now instead of going from 0% to plus 100%, we're going from about 4 notes per second to about 24 notes per second. So the quick answer is no, it is not absolute tempo that makes this the most attractive thing she's ever heard. Instead, it is a relative change in tempo that is the best predictor of any of these features that we've looked at. So this knowledge from Bengalese finches is really good because this is such an experimentally tractable species. We can go in and we can ask more high-resolution behavioral questions. We can ask neurophysiological questions. And it allows us to understand our data excuse me, better in light of what's come before. That here's the swamp sparrow where what you do, the impressiveness of your frequency bandwidth, and the speed at which you do it are interrelated. And it is not a simple high bandwidth that makes a song attractive. It is not a simple high tempo that makes a song attractive. It is doing something rapidly that is physically demanding. That's what makes this song attractive. And it turns out when we look higher, more detailed at the canaries, these sweeps must go from 6,000 hertz to 1,000 hertz. If they don't, then it won't be recorded as sexy by these females. And these trills must occur at least 15 notes per second. If they don't, then the females don't find them attractive. So again, you have to do something that is physically challenging, sweep from 6 kilohertz to 1 kilohertz, and you have to do it rapidly. And then females, when they hear this, find this song attractive. So this is now, from a practical point of view also, a tool that we can start to use to make a song more or less attractive based on what we know about this female. Do you respond to changes in tempo, and can we use this as a means of making a stimulus more or less attractive for you? And that allowed us to begin asking how signal value and mate choice are encoded in neural activity. So we've just begun these experiments, but we can think back to what we saw about the male bird. And the male bird brain contains this red path that's necessary for singing, and this blue path that is associated with perception and song plasticity. And in the species that we study, the males can sing and the females cannot. So the females not being able to sing means that we can effectively take those pathways away because they're so strongly atrophied. And we can have auditory inputs projecting to the primary auditory cortex, then to these secondary or associational auditory cortical areas, and finally up to this structure called shelf, or HVC shelf, which is a very high order processing center. And field L is itself not that interesting to us because it is the primary auditory cortex. It's so broadly responsive. It responds to songs and my voice and twig snapping and pure tones and white noise and everything. CM, on the other hand, is interesting to us because that lesions in CM can alter mate preference. If you go in and physically destroy part of this structure, then preferences which were previously restricted to birds of your own species become quite promiscuous. You're now responsive to multiple species. And immediate early gene responses to the mate song and other songs that the female finds attractive are quite strong. And an immediate early gene response is something that tells you that these cells have been active. So CM is very strongly activated in response to these songs that behavioral tests tell us, or behavioral observations tell us are important. NCM is responsive to the mate's call. It was recently published that electrophysiological activity is selective for some cells for the call of this bird's mate. And again, there are strong IEG responses to attractive songs. And HVC shelf, lesions alter mate preference. There's some who believe that was actually lesioning CM because the lesions were so large. But some say HVC shelf matters. We'll take a broadly expansive view of it. But we also know that, that auditory activity in CM and NCM is altered by lesions in this site. So it appears that we have sort of this triad of areas that are interconnected and are conspiring to contribute to the evaluation, first of all, recognition of one song being associated with my mate versus not, and second of all, evaluation of the quality of this signal. So what we're considering now in the lab is in what larger context do these areas reside in order to affect motor behaviors? Because I can make you think that a Big Mac smells good, but it's another thing entirely to make you open your wallet and give me money for it, right? To execute the associated motor decision. And then we're asking, how is it that preference and mate choice are encoded at the level of individual neurons? We're trying to understand this. It's still early days, and hopefully there are a lot of opportunities for collaborations between Sarah, John, and our lab. So in summary, we've seen that these male Bengalese finches express preference for individual females. They are evident not only as the properties of the songs, but in addition to the number of songs that they perform. They're functionally relevant and they influence the evaluation of the females for what they believe is the quality of this song. Then the females express preferences for individual males. It's evident in both their copulation solicitation displays, which is this behavioral bedrock that allows us to understand that it is also evident in the number of calls that they perform. And it appears to be very strongly related to relative, or most strongly related to relative changes in song tempo. So we're pleased that we've got now this behavioral bedrock to go into the neurophysiological investigation of how this signal evaluation occurs. So I'm grateful to graduate students in the lab, Kara Murphy and Jeff Dunning, who have been doing this. We've had a lot of talented undergraduates come through. I'm grateful to our collaborators, Luke Ramashvili and Todd Roberts, and of course those that pay the bills. I'm grateful to you guys for the chance to visit, and I put this picture up because this is about 15 miles from our house. If you're ever down in Wyoming and you want to come for a similar hike, I'd be happy to take it. Thank you guys.
Questions?